Thank you very much indeed for the kind introduction. It's great to be here this evening. I'll probably be upstaged by the Aurora Borealis later on if you go outside at home, because there's going to be a sighting of it down here later on. But hopefully I'll be able to fill the gap over this next hour. And I wouldn't be here this evening were it not for this gentleman here. This is a published photograph. And I met him in the mid-1990s when I was training as a junior neurologist in Newcastle upon Tyne, and he was admitted, as people were in those days, for a full week to try and help us make a diagnosis, to understand the cause of his problem. And during the course of the week, he had a number of clinical tests carried out. You can see actually that he's lost a lot of weight because he can't swallow properly. He was very clumsy. The neurological term for that is ataxia. And you can see on his brain scan the balance organ, the cerebellum at the back of the brain, has shrunken prematurely. He had a problem with conduction in the heart, and this is an ECG showing that heart rhythm disturbance. And he was profoundly deaf, as shown on this audiogram. This resonated with me because he was born the same month that I was born in, and we had a very different prospect ahead of us. Unfortunately, he's long since deceased now, but he set me off on my course. And towards the back end of the week, we got insights from a muscle biopsy, and this is a picture of the muscle biopsy here. Each one of these hexagons, rough hexagons, is a muscle fiber. And what the muscle fibers show is a collection of particles underneath the surface of the membrane of the muscle fiber, giving us a clue as to what the diagnosis was. And he had a problem with these. These were first described by Carl Benda in 1898 as thread-like granules. And for the Greek scholars in the audience, mitochondria. And they're the focus of the talk this evening. So what are mitochondria, and what is it that they do? Well, they're present in every cell in our body, and this is a stylized version of the cell. You can see the nucleus containing most of the genetic material and the various compartments here. And the mitochondrion is shown here, and you'll see there are several inside each cell. And if you blow them up, you'll see the structure has two membranes, an outer membrane and an inner membrane. And the inner one is folded over many times, giving you a clue that the surface area is important. This is a place where chemical activity occurs. And mitochondria are the main source of energy in cells in the body, particularly when we're in an oxygen-rich environment. So why, if it's not obvious, are these important, and how did they arise? Well, to understand the origin of mitochondria, we have to go right back in evolutionary time. This is a tree showing the evolution of the species, with us down here at the bottom, a rather late arrival on the scene. And if you look back in the fossil record, hundreds of millions of years, you'll see something that's described as the Cambrian explosion. It's a huge diversification of species. And for that to happen, you required high amounts of energy to move beyond organisms that were single cells to complicated multi-cell organisms, and ultimately to mammals and humans. And mitochondria were an essential step in that journey. And the prevailing view is that a very long time ago, in fact, it's thought at present to be about 2.5 billion years ago, a primitive cell associated with some bacteria. Some of those resembled cyanobacteria, and these went on to form plant cells. But both plant and animal cells took in something resembling an aerobic bacterium, a purple bacterium resembling rickettsia. These are organisms that are around today. And it established a symbiosis with the cell, a symbiosis that has endured to this present day. And the mitochondria took oxygen from the cell and in return gave it a form of energy. And that's how the symbiosis works. And we've learned an enormous amount about the machinery of this over the last 30 to 40 years, not least from my colleagues here in Cambridge. And if you want to get into the detailed machinery, this is a picture here showing the inner membrane, inner of those two mitochondrial membranes, and a group of over 100 proteins that are assembled into enzymes, which are chemical catalysts. And these are numbered 1 to 5, and they're involved in this process of respiration and the production of energy. How does it work? Well, the breakdown products of the food we eat feed electrons into complex number 1 and number 2. And these electrons are passed down between the various complexes in this so-called respiratory chain and eventually are tagged onto oxygen. And in doing so, you make water, which is one of the byproducts of this chemical reaction, and you pump hydrogen ions, protons, from inside the mitochondrion into the space between the two membranes. This creates an electrochemical gradient across the inner membrane, and that is harnessed by complex number 5 to drive the synthesis of adenosine triphosphate, which is the high-energy molecule that we need for all active cellular processes: muscle contraction, nerves firing, protein synthesis. They all need adenosine triphosphate. And the molecular basis of this has been worked out, and you can see actually the diagram's not quite complete because we now have a structure for complex 1. And how it works has been worked out in large part by colleagues here in Cambridge. And one of my close colleagues, the Nobel laureate John Walker, worked out how complex 5 works. And here it's turned up the other way around. So this is the inside, this is the intermembrane space between the membranes, this is the matrix. And when the protons are flowing back from across the gradient, they drive this rotatory action of the ATPase. And by driving that rotatory action, they expose sites on the rotating head here, which expose the raw materials that form ATP to come together and to form these new ATP molecules. This is happening 7,800 times per minute for each one of these. And in the last 24 hours, you have replaced your body weight by making ATP using this mechanism. It's extraordinary. And in a way, it's amazing that we're here to tell the story. It's so complex. But as a neurologist, I've spent a large part of my career identifying people where their mitochondria are not working properly. And this, of course, has an impact on many different organ systems, as in the patient I described to you right at the start. Typically, these conditions can affect the nervous system, shown on the right, but they can also affect other parts of the body, the gut. They can affect hormone production, they can affect the liver, they can affect the heart. And it's really these constellations of clinical signs which give us a clue that there's a problem with the mitochondria. And we've actually learned an enormous amount about how mitochondria are synthesized, put together, by studying families and patients similar to the one that I described to you at the start. In a way, the patients that we've seen over the last 20 to 30 years are a bit like the individuals who suffered gunshot wounds in the First World War. In that context, gunshot wounds caused a lesion, a problem in the nervous system, which told us exactly what that part of the nervous system did in health. But exactly the same in people with mitochondrial disorders, because by studying and characterizing the underlying molecular basis of these conditions, we get a handle on what's important for mitochondrial function in health, and of course its relevance to disease. So we now know that mitochondria are made up from about 1,100 different protein building blocks, and that these are coded for by a genetic material in the cell nucleus, the chromosomes in the nucleus, for which there are 2 copies. But the intriguing thing is that mitochondria also contain their own genetic code. Small circles, which you can see in this figure here, called mitochondrial DNA, or mtDNA for short, and that's going to be the topic of this lecture this evening. Now, the interesting thing from my perspective is that the respiratory chain, this group of proteins that makes energy, requires both genetic systems to work in concert. You'll recall, of course, that most of our genetic material is inherited from both parents, part from our mother, part from our father, going back over the generations. But our mitochondrial DNA is only inherited down the maternal line. All our mitochondrial DNA comes from our mother. These 2 genetic systems never physically touch each other, but they need to work in concert for our mitochondria to work properly. And what we found over the last 2 decades is that patients with mitochondrial illnesses that often come to the neurology clinic can be caused by spelling mistakes or mutations in the genetic code that comes down the nuclear line from mother and father, or down the maternal line in the mitochondrial DNA. Now, early in my career in Newcastle upon Tyne, I had a long-standing mentor, Douglas Turnbull, shown here on the left. We spent time identifying people with these conditions across our region and characterising them as best we could with the tools we had at the time. And it allowed us to get a handle on how common these conditions were. And actually, this data has stood the test of time. When I first went into the field, these were thought to be exceptionally rare, perhaps a handful of people with these problems in the country. But it turns out that they affect about 1 in 8,000 people. Now, that puts these conditions along with other conditions you may have heard of, like Huntington's disease, for example, a similar frequency in the population. And we also found that in the adult patients we studied, about 2/3 of the individuals, just like the man I told you about at the start, had mutations or spelling mistakes in these small circles of DNA, mitochondrial DNA. And so I'd like to talk a bit about this molecule. So this is mitochondrial DNA. It's small. It's only 16,500 letters or base pairs long. And as far as we know, all it does is code in these different colors for the proteins, 13 of the proteins that are essential for this energy metabolism, the rest of the 100 coming from the nuclear genome. And the other bits of this circle are part of the machinery the mitochondrion needs to make those proteins inside itself. Now, there are many copies of mitochondrial DNA in every cell, typically thousands, and in some cells, 10,000 or more. And so if there's a mutation or a spelling mistake, it causes a mixture, and that mixture has a special term. The term is heteroplasmy, and it's important. We'll come back to it a little later on. But the point here is, and it's shown in this cartoon, if the red is the mutated or mutant form, that the mixture or the proportion of heteroplasmy can vary over a range of 0 right through to 100 nearly. And that's important because we now know that you have to have very high levels or high proportions of a mutation for it to affect the function of the mitochondria in a cell. And there's a characteristic threshold level which is typically very high. 80% in a cell has to be affected before the cell doesn't work properly. And these are important concepts we'll come back to in a short while. Now, these diseases can be horrendous. This is from the Sunderland Echo. It's a family that I looked after for many years. And the family described this cruel family curse that runs down the maternal line and the difficulty of making any predictions as to whether it's going to affect the next generation. The family actually came to our attention quite late when there'd been a lot of problems because of an important clinical point I'll make. Some of the individuals had problems with their heart muscle problem. Some were deaf. Some had epilepsy. And there was lots of mid and late trimester or late pregnancy loss, spontaneous abortion in the pregnancy. And the key point was that each different person had a different problem, so no one put the jigsaw together to think this was a genetic condition. They just thought these people in this family had randomly got different illnesses that by chance had come together. But we now know this is an inherited disease. And the question is, how does it arise? And why is it relevant to the topic of tonight's lecture? Well, to understand how this arises, one has to think a little bit about the way the mitochondrial DNA is copied in the cell. So this is a cartoon of a cell. This is the cell nucleus. This is a mitochondrion. Here are the 5 respiratory chain proteins sat on the membrane. These are the circles of mitochondrial DNA. And the interesting thing about mitochondrial DNA, which is different to the cell nucleus, is that it's constantly being destroyed and copied. The copying is carried out by an enzyme, a catalyst that copies DNA, that comes from a gene in the nucleus. And it's doing it all the time, even in cells that aren't dividing, such as your brain cells and muscle cells. And this can happen up to 10,000 times in one of the neurons that's in your brain. And so it introduces errors. And you can see these errors accumulating over time. And for that reason, mitochondrial DNA is thought to have a very high mutation rate. So if that's happening, can we see evidence of this in healthy people, such as those of us sat here in the audience? And for that reason, Vasilios Floros, who was a junior scientist working in my lab, decided to look in the germline, the female egg cells, to see whether or not we could detect evidence of these mutations of mitochondrial DNA that we would predict would be there because the molecules are turning over all the time and errors happen. And so he developed new techniques to isolate germ cells, the future egg cells, from female embryos. This is one of them here. It's tiny, it's only 5 microns across, and the mitochondria are staying green. And he took the mitochondrial DNA out of these cells, and he sequenced the mitochondrial DNA in each cell. And the graph shows you the summary of the experiment. So along the bottom, this is the mitochondrial DNA here, drawn out into a long strip from its circle. And each spot is a different cell that was sequenced many, many times. And the take-home message from the data is that actually almost all of these healthy egg cells in these healthy embryos contained mutations of mitochondrial DNA, different ones in each cell. But you can see the proportion affected was quite low, only a few percent. So what this is telling us is that mitochondrial DNA mutations are out there in the audience, and they're in the germ cells that become egg cells in women, and this would predict that they are passed on from generation to generation. So the question is, can we see that in the general population? Well, Wei Wei, who's here tonight, did some key work to show that this is indeed the case, and she took advantage of 2 studies here in England where large numbers of people had their whole genome sequenced, part of the 100,000 Genomes project, and some of you may have been involved in it. Part of it was coordinated by an organisation here called the NIHR BioResource, which was the pilot for the Genomics England study. And she looked at 53,300 human genomes. Now, how is that helpful? Well, this cartoon explains why. So this is a picture of a cell again, cartoon. You can see the mitochondrial DNA, you can see the DNA in the cell nucleus. And when you sequence a genome, the first thing is to smash the cell apart and take the DNA out of the cell. And this process doesn't separate mitochondria and the nucleus, you get a lot. And when you sequence the human genome, you sequence everything that's been taken out of the cell. And the system's set up deliberately so that when you sequence the nuclear genome, you sequence it about 40 to 50 times. Why would you want to do that? Well, you'd want to do it because if you find a spelling mistake, a mutation that might indicate someone has a disease, you want to be very sure that that change is real. So you do it 40 to 50 times. Now, because there are many, many more copies of mitochondrial DNA in the cell than nuclear DNA, then for free, as it were, you also sequence the mitochondrial genome, but over 1,000 times. And that allows you to look for heteroplasmies, this mixture of mutations and normal, so-called wild type, down to the level of about 1% in the sample. And so Wei set out to look at these in these people in the 100,000 genome projects. I'll just tell you a bit of the information here, focused on trios, which are a mother and 1 or 2 of her children. She looked at 1,500 of these trios to look and compare the mother to the child and looked at the mitochondrial DNA. And the information I'll show you is going to be broken down into 3 bits. Mitochondrial DNA mutations that are transmitted or inherited in gold, in blue, ones that are lost. In other words, they're present in the mother but not in the child. And then the converse, de novo, in other words, they're not present in the mother but they're present in the child, they appear. So what did Wei find in these 1,500 mother-child pairs? Well, this is what she found. Let me walk you through it. Again, here's the mitochondrial genome cut into a long strip. Just ignore the axis. It's a mathematical computation. It doesn't really matter today. But the striking observation is there is an awful lot going on. So in these 1,500 mother-child pairs, each one of these arrows is a mitochondrial DNA mutation, which is either transmitted, lost, or occurs de novo. It's a new event. There is an enormous amount of diversity. In fact, half of the trios were found to have these mutations with this threshold of 1%. Now, if you take all that information and you reorder it from the biggest shift down to the biggest shift up, you just order it in rank order from the biggest shift down to the biggest shift up, this same information looks like this. So in the middle, the gold are the ones that were passed down either from the mother, and they're detectable in the mother or the child. Here are the ones that are lost, they go down. Here are the de novos that appear, they go up. And what we can see in this list, if you're an aficionado, is that some of the changes are really quite dramatic. A 64-fold change in the level of the mutation down just one generation, heteroplasmy changing. And in that little list, we can see some of the ones that we recognize as causing diseases in the patients we see in our clinic. So it's telling us these mutations are out there and that the level is changing between a mother and a child, and occasionally this can pop up to high levels exceeding the threshold, causing the illnesses, as in the patient I described to you earlier. Now, from a mathematical perspective, you can plot this data, tip it on its side, and plot a distribution where 0 is no change, and this is up, and this is down. And what you'll notice on that distribution is it's broadly symmetric. Levels can go up, and levels can go down. How on earth does this happen? Well, this has intrigued me for 2 decades, and I think we have some insights into what's going on at the cellular level. And let me explain it to you. It's called the mitochondrial genetic bottleneck, which was first thought about 50 years ago from observations in heteroplasmic cows, where changes in heteroplasmy were observed down several generations of these cows. And we see the same thing happening in most species. And the key events we and others have been involved in dissecting them out over the last 2 decades, and it all hinges on a very early stage in development in females, in women. So early, in fact, that to get a handle on it, you've got to study the steps from fertilization through to the ball of cells, the so-called blastocyst that sticks on the wall of the womb and starts to develop an implant. And shortly after the blastocyst sticks to the wall of the womb, then the next generation of germ cells, egg cells, start to be formed, and these are called primordial germ cells, and these proliferate and form the ovary containing the oocytes, which are the egg cells for the next generation. So all this happens in a very early stage in embryonic development. And what we've shown that at this critical period of time, around the point that the primordial germ cells appear, there is a genetic bottleneck that occurs. What do I mean by that? What I mean is that the amount of mitochondrial DNA in these primordial germ cells drops to a very small amount. And this causes a statistical sampling effect. How does it work? Well, imagine I had a big bowl here full of, in this case, blue and red balls. Imagine I closed my eyes and put my hand into there and pulled out a selection each time. Now, if I had enormous hands, took a huge sample out, then each time I did it, it'd be more or less the same. But if I only took out a small amount each time I did it, you get very different proportions of the red and the blue balls. And that's what we've shown is happening during the germline development in terms of your mitochondria. And what it means is that the level of heteroplasmy, the proportion of the blue and red, can change in each one of these, change during this process in each one of these cells. And sometimes the level can go up to very high levels and cause the disease. And we've measured this in mice and latterly in humans and shown that depending on the species, hundreds of thousands of molecules in the fertilized egg goes down to a few hundred in these germ cells. So this explains why heteroplasmy can shift between one mother and her child. Can we see evidence of all these mutations in the population at large? And we also looked at this a few years ago, looking at 30,000 mitochondrial DNAs taken from all around the world. And this diagram shows you what was discovered. So the circle, as you'll be familiar now, is the mitochondrial genome, and each inner circle represents genetic variation, polymorphisms, changes in the genetic code in different parts of the world. And the take-home message is there is an awful lot going on in each different part of the world. Mitochondrial DNA is highly polymorphic. And if I was to sequence mine and compare it to yours, there would be about 60 differences between the two. That's a lot of differences for a small genome that's only 16,500 base pairs in size, much more than you get in the nucleus. And of course, these genetic variants are all ultimately can be traced back to a single woman, theoretical Eve, who arose out of Africa 150,000 years ago, and her ancestors down the female line went out and populated the world, and as they moved around the world, they collected these mutations of mitochondrial DNA, and because it only is inherited down the female line, this causes a great tree down the female line, and that the genetic variants or polymorphisms occur as we move into different parts of the globe, and this information has been used by the anthropologists, the population geneticists, to infer how humans populated the Earth. And in doing so, led to a number of groups called haplogroups, which we'll come back to in a short while. So here we are halfway through mitigating mitochondrial mutational meltdown. We've got a problem. Here's a system where the genome's constantly copying. Errors happen because it's happening all the time. There's a system that can change the level down one generation, pushing high levels of mutation up between a mother and a child, and you can see all this genetic variation at the population level. This problem was recognized nearly a century ago by Hermann Muller in this article. I've put the DOI at the bottom because the article is called Some Genetic Aspects of Sex, and it's in a journal called The American Naturalist, and I wouldn't recommend you Google any of those terms, at least not on a university computer. Now, Hermann Muller, in this opening paragraph, says, from the genetic point of view, it is advantageous to begin by considering sex in the broader sense of sexuality. It is not generally realized that genetics has finally solved the age-old problem of the reason for the existence, i.e. the function of sexuality and sex, and that only geneticists can properly answer the question, is sex necessary? There is no basic biological reason why reproduction, variation, and evolution cannot go on indefinitely without sexuality or sex. Therefore, sex is not, in an absolute sense, a necessity. It is simply a luxury. Now, there are all sorts of issues raised in this paragraph, and this clearly reflects a different era. We could go on talking about some of these elements well past midnight. But in the same article, Hermann Muller very astutely recognized that the reason why we have evolved to reproduce sexually is to limit the accumulation of mutations. And he proposed something called Muller's ratchet, which applied to any form of genetic system where it was inherited just down one parent, like mitochondrial DNA. And he recognized that if you have such a system, then it will relentlessly accumulate mutations over time and eventually lead to extinction of the species. So can we save the species? Well, let's begin with the patients with rare inherited mitochondrial diseases, because colleagues in Newcastle, team led by Doug, you'll recall 4 or 5 years ago obtained a license for a new treatment, and this led to a great press furore about babies with two mothers. And the reason why that was suggested was because of the way the treatment works. And the idea behind it was that you could effectively use mitochondria that have been donated by a woman with healthy embryos to swap any inner embryo that are coming from a mother carrying a mitochondrial DNA mutation, so that you only pass on the nuclear DNA from the mother with the mitochondrial DNA mutation. You leave, if you like, the bad mitochondria behind and bring in the newly donated healthy ones to form a healthy embryo. So-called mitochondrial transfer therapy, which is a therapy that's been done 4 or 5 times in the world and is a licensed therapy on the National Health Service. So this work provides some hope that we can prevent the transmission of these problems in families. But it can't be the whole solution. My first PhD student, Hannah Elliott, carried out a study in west of Cumbria, and she looked at 3,000 sequential live births in Whitehaven Hospital, where the women, perfectly healthy, had given a bit of the cord blood for analysis. And she looked to ask the question, how common are mitochondrial DNA mutations that cause disease in the population? How common are they? And she found, and it's a long time ago, 2008, that about 1 in 200 of these women had detectable levels of disease-causing mutations at low heteroplasmy levels in their blood. And Wei did similar work on the 100,000 Genomes Project and came up with a more sophisticated result of about 1 in 250 by sequencing the mitochondrial DNA. And together, these show that these severe mutations that cause disease happen de novo, new, fresh, every 1,000 births. So this is going to happen all the time. Doesn't matter how good we are at getting rid of them, they're going to keep coming back in. And unfortunately, it's a bit more complicated than that too. So I'd like to cast your mind back to the tree the tree I showed you descending from mitochondrial Eve, of genetic variants of mitochondrial DNA in the healthy population, so-called haplogroups. And this describes some work that we recently did in partnership with Joe House and Kate Doing and Claudia Calabrese from my group, where we looked at the mitochondrial genomes in 350,000 people in the UK who become participants in a big national study called UK Biobank. And we characterised the mitochondrial DNA in these 350,000 people in great detail. And you can see here all the different types of mitochondrial DNA that we looked at. And this shows you the distribution of the different types all around the country. Really interesting, because it allows us to also make some inferences about population migration. For example, we could see the signature of our friends in Scandinavia who came to the UK around 800 AD. The genetic signature of the Vikings is there. But the reason why we were interested was to see whether or not these genetic variants that you and I have are important for common diseases as well as the rare ones that I see in my clinic. And the answer is they are. We correlated the genetic variants of mitochondrial DNA with traits that had been measured in UK Biobank participants. And we found that mitochondrial DNA, your mitochondrial DNA, influences 227 of these traits. And we found associated with things we just couldn't have imagined. What this diagram shows you is it shows you the genetic association, again, of bits of mitochondrial DNA, the circle, with traits shown like different parameters in your blood count that the GP carries out, the platelets that are involved in clotting, circulation, and also the endocrine system. And the figure shows the measure of genetic association between mitochondrial DNA and these parameters. So things like your liver function, your kidney function is partly influenced by your mitochondrial DNA. And it also influences your risk of common diseases like multiple sclerosis and type 2 diabetes. And even more interesting than that, some of the genetic variants also influence characteristics that you might not think were genetic necessarily. So this particular variant here affects your height on average by about 4.3 millimeters. Your mitochondrial DNA is influencing your height. Tiny amount, but this is as strong an effect as any other gene in the nuclear genome. Or this one. If you have this particular genetic variant, you live on average 30 days longer. Very useful for me this week because I'm doing the Cambridge Half Marathon on Sunday, and an extra 30 days would be very useful training for me. So our mitochondrial DNA doesn't just cause these rare diseases, it influences who we are, what our physiology is, and thereby some of the common conditions that we may be affected by. What are we going to do about this? Well, if you look at the treatments that we can offer our patients in the neurology clinic, here's the list. Don't read it, because if you ever see a list of treatments that's extremely long, it's a good guarantee that none of them particularly work. And that's the case for mitochondrial disorders. We really don't have any convincing treatments for these conditions. And the question is, can we redress this and put this right? So in the last quarter of an hour or so, I'll share some recent insights with you that I think are moving us in the right direction. And it comes, of course, from the patients that we see in our clinic. And the question is, what can we learn from these people? And although, as doctors, we tend to focus on the parts of the body that aren't working well, also we can learn from parts of the body that are working well. And the question is, are there things about the organs that are not affected that are also going to turn out to be important for developing new treatments? What is it that protects some organs, given the fact that mitochondria are so important for every cell in the body? So I'm going to tell you a bit about some work done by Stephen and Angelos and Malvina, two of whom are here this afternoon. And the first step is to ask the question, are mitochondria all the same in every part of your body? Very difficult question to solve. A little clue from the protein literature that they might not be, but we've taken advantage of some new technology to cast light on this. And before we get into it, I just want to remind you, of course, that all DNA, mitochondrial DNA and nuclear DNA, is transcribed into a message called messenger RNA, and that is translated by the protein machinery, the translation machinery, to make proteins. They're the steps of protein synthesis. And we've taken advantage of a new technology that really is transforming medicine and biology at the moment, which is to look at the transcripts, these molecules, in single cells at scale. To do that, we've started with mice, and we've looked at a particular stage in early development of the embryo when it's about 2/3 of the way through at 8.5 days, and collected the whole embryo and isolated 5,000 cells selected at random and sequenced all the transcripts, all the messages produced from the DNA in each one of those cells. Very powerful. So what did we find? Well, this is a way of representing the data. It doesn't really matter how it's done, but essentially it's a statistical method of clumping data that's similar and separating data that's different. And this is, of course, based on the transcripts or the messages produced from the DNA in each individual cell. And when you look at that, you can see that the cell groups tend to separate out in different forms. And we can paint them, which is effectively what we've done, with different colours based upon specific messages or transcript that we know already are the signature of a particular cell type. And you can see these cell types here. So, for example, this lot here are all different parts of the brain, the forebrain and the hindbrain, and they're related to the neural crest, which is one of the embryonic precursors of the brain. So this is a very powerful technique, and each dot there is a single cell, and it allows us to count the number of cells of each type in these embryos. And the really interesting observation is shown here. What this shows is 4 different messages, transcripts, and it shows them across different tissues. And what you'll notice is that some of them appear more strongly in some tissues than in others. And the one I'd like to draw your attention to here is cardiac, that's the heart form. All 4 of these transcripts code for parts of complex number 4 in the machinery, the respiratory chain I described to you earlier. And what you'll see here is that these 2 here are only expressed in heart cells. These are isoforms. They are genes that code for specific proteins that are coded for that go into the mitochondria that are only found in heart cells. So it's remarkable. Even before the heart's formed, we can pick up the signature of specific mitochondria for a particular cell type, almost predicting what the requirements will be later in life. So the answer to the question is all mitochondria are not the same. So what happens if we introduce a mitochondrial disease, a bit like the patients I've described to you earlier, this time with the work of Jim Stewart, one of our collaborators now in Newcastle, 2 different models which resemble human diseases caused by spelling mistakes, mutations in this particular structure here called a transfer RNA gene. It's not important. But when we look at take the same approach using these disease models, we can see when we look at the data that there are differences. So what this busy slide shows you is it shows you each little stripe is a single cell, and down here you can see some of the different genes coding for different parts of the mitochondria in the cell, and you can see the healthy mouse, and these are the two mutant mice. And essentially what this shows you is there's a pattern. There are differences between these different mice in the transcripts, these messages, at the single cell level. So what's this telling us? Well, to interpret this, we went to the literature and some work that came out of a collaborator's lab, Vamsi Mutha, in Boston. And he did this a couple of years ago. He did a very clever experiment. He took cells in a culture and he poisoned the mitochondria with very specific chemicals. And he let them grow. And what he did was he sequentially, across the whole human genome, knocked out each individual piece of genetic code. And he looked to see which one of those disrupted genes helped the cells or hindered the cells in the context of a mitochondrial poison. And so in doing so, he identified a number of genes which, in the context of a mitochondrial insult, are lethal or beneficial. And we hypothesized that we might see the same thing in our mice with these mitochondrial diseases. We'd predict that in our mice that these genes may be upregulated in the mice, or these may be downregulated to buffer the effects of the mitochondrial mutation. And to cut a long story short, that's exactly what we found. There's a very strong enrichment for these buffering genes, and this is a bit of the information shown here. So this is the normal mouse. This is one of our disease mice. This is another disease mice. This is a list of different genes. The green ones are mitochondrial proteins. And what this is telling us is that there are certain genes which are downregulated in the context of a mitochondrial insult in a tissue, and this is also in a tissue-specific way, as shown on these plots, similar to those I described to you earlier. What these two plots show is they show you some master gene regulators. In this instance, one called MECOM, which is downregulated in this cluster of cells called the endothelium. And on the right, another master regulator called SOX9, which in this case is upregulated in the cells that are going to form the neural crest in the context of the mitochondrial disease mutation. So this is giving us the first insight into mechanisms that explain tissue specificity, and we hope this may also give us some insight into targets that we might work on to develop new treatments. And this was published just last Thursday and is available online. So to conclude this bit, as we're approaching the end of the lecture, I've talked to you about mitochondria, I've talked to you about their essential role in life and how genetic mutations affecting the way we make our mitochondria can cause a whole host of different problems that present to our clinic, but that these overlap with the common conditions like Parkinson's disease, multiple sclerosis, type 2 diabetes that you'll be much more familiar with, and that our work in UK Biobank gives us little insight into common mechanisms. Now, I'd just like to change tack for a final little story before we wrap things up this evening and have a discussion, and go back to this slide that I showed half an hour ago. And it summarizes how mitochondria are made from the products of two distinct genetic codes, one of which is exclusively transmitted down the maternal line. Now, this has been known for 60 years in mammals, exclusive transmission of mitochondrial DNA down the female line, but actually it was questioned a few years ago by colleagues in China and North America, and this actually amazed me because it hit the mainstream press. I'm delighted to see that the Daily Telegraph and the Times and the Guardian were interested in mitochondrial DNA. But what the colleagues here reported was that the mitochondrial DNA was not just being passed down the mother's line, but also being passed down the father's line too. A really radical change, which goes right back to what Hermann Muller was telling us about uniparental inheritance. And this is the data. So this is from their paper. And it shows the mitochondrial DNA in different generations of the family. And each little number is a position on the circle of mitochondrial DNA. And each bar represents the proportion of mitochondrial DNA that's affected by that genetic variant, so 58, 100%. So you can see there the great-grandfather and the great-grandmother, they're different, they're not related. But if you look at the grandfather here, so their offspring, there's a mixture. And there seems to be a mixture of the great-grandfather and the great-grandmother. You can see them stacked on top of each other here. And it was this, on this basis, that they reported paternal transmission. Interesting now that you've heard this lecture, that you'd spot there are some curious things about this, because multiple sites were affected, and the level of the variant, the heteroplasmy, was more or less the same in each instant. It looked as though 40% had come from the great-grandfather and 60% had come from the great-grandmother. And even more amazing, it was passed down another generation. And so this is how they built their case. So we set out looking in the 100,000 Genome Project for similar examples, and again, largely the work of Wei Wei. We looked at 12,000 trios, so little mother, father, child, and said, can we see evidence of this paternal transmission of mitochondrial DNA? And in 7, we saw the same signature. Here it is. This is family 6. Here's the father, here's the mother. You can see in one of them there is a mixture of the father and the mother, and here you are. It's in the sibling too. Very similar to what was described in this revolutionary paper. So what do we think is going on? Well, I think the reason why I put this story at the end is because it takes us right back to our understanding about mitochondria, how they arose, and how they are evolving. Because one thing I didn't tell you is that over evolutionary time, this symbiosis has evolved to such an extent that some of the functions of the mitochondrion have been passed to the host cell. And that's been achieved by mitochondria transferring bits of genetic code into the cell nucleus. This is the cell nucleus. These are the mitochondria. So you can see bits of mitochondrial DNA going in and sticking inside the nuclear genome here. And these are called nuclear mitochondrial sequences. And they've been known about for a long time. In fact, if we look at your genetic code, you'll find 755 bits of mitochondrial DNA stuck inside your nuclear genome. In fact, the whole mitochondrial DNA sat on chromosome 1. So we've known about these. These were thought to have happened a long time ago in the ancient evolutionary time. But we wondered whether this could be the explanation for what the North American group had misinterpreted as being paternally transmitted mitochondrial DNA. So it's a bit late in the evening, but I promise you we'll finish on time. But I need to talk to you a bit about some technicalities, if you can bear with me just for a few more minutes. And that is to explain how it is that we sequence the whole human genome. We do something called paired-end sequencing. It's quite clever. When you break up a chunk of human genome, what you do is you sequence it twice. You sequence it once at that end and once at that end. And by comparing those to the reference genome, the known reference, you can infer what's sat in between and thereby build up a sequence across the whole genome like this, using these long fragments in something called paired-end sequencing. It's done for a technical reason, really, to try and help pick out repeat sequences, which isn't important today, but it's very important because it gave us a tool to ask this question: are the mixed haplotypes that we see in these trios due to real paternal mitochondrial DNA transmission or something through the cell nucleus? And so we established a pipeline to look at this, and I'll just walk you through. Here's the mitochondrial DNA. Here it is, inserting into the nuclear genome. So this is what you might imagine it looks like. And these are the paired-end reads that we routinely use to sequence the human genome, color-coded to make it a bit easier. And we set out to look for what are called discordant reads, in other words, paired reads which match partly to mitochondria in orange and partly to the nucleus in blue. And also split reads, where part of the bit you actually sequence sticks to the nucleus or to the mitochondria. And you can use this to detect these theoretical mitochondrial inserts. And we found them. So this shows you the father, the proband, sibling, and the mother from one of the families I described earlier, family 2. And what the colours show you is it shows you where these reads map to on the nuclear genome on this side and the mitochondrial genome on this side, chromosome 13 on the left. And you can see the paired end reads mapped to both regions, telling us that there is a mitochondrial DNA insert that had not been described before. And this diagram shows it in detail for you. Here's the nuclear genome, here's chromosome 13. This shows you the coordinates mapping onto the mitochondrial genome here. So what we found is that there were some very large nuclear mitochondrial sequences stuck in the paternal genome that had never been described before, that were being passed down, and that whenever we found these, it was 50-50 whether they'd be passed on to the offspring. So what this is telling us is that, at least to our mind, there is no reason to change the dogma that mitochondrial DNA is inherited down the mother's line, and that what you might see is pseudo-heteroplasmy arising because of bits of mitochondrial DNA going into the cell nucleus. Encouraged by this, we did this on a bigger scale. And Wei and Catherine, who's also here, have now looked at this in more detail. We've done some technical work to prove that this technique we're using works by doing very long read sequencing that goes on for much longer than the short reads that we routinely use. And Wei's looked at 66,000 human genomes and also some cancers, comparing normal tissue to the cancers. And what did she discover? Well, she discovered extraordinary variability of these. What this is the summary of the data. These are the nuclear chromosomes. This is the mitochondrial DNA. Totally unanticipated diversity in these nuclear mitochondrial inserts. Almost everybody's different. These map to the nuclear chromosomes, which are shown here. Each one of these yellow bars is a bit of mitochondrial DNA that's found its way into a nuclear genome in a different person. 99% had one or more completely new mitochondrial inserts. 1 in 8 had very rare ones that presumably have happened really recently, and most of them had never been seen before. No one had ever looked. So what about the cancers? Well, there's less information, so the diagram's not quite as pretty, but effectively it's the same story. Nuclear chromosomes, mitochondrial DNA. What we're finding is cancer genomes are stuffed full of new chunks of mitochondrial DNA, and it varies from cancer to cancer, bladder cancer being the worst, some childhood cancers having the least of these. Extraordinary diversity, more than you see in the blood. Different cancers are different. And what this is telling us, not only is this happening as our genomes are being passed from parent to child, but it's also happening in our tissues as we sit here. Sometimes in cancers, sometimes in normal tissues, somatic tissues, bits of mitochondrial DNA are passing into your nuclear genome, even as we speak. And we can measure how frequently it happens. This is one of the families. It happens, we can detect it about 1 in every 4,000 live births. It's happening all the time. So you've been very attentive, and I'm very grateful to you. I presented this model to you as to how mitochondria are made. It's a tale of two genomes. And the way I presented it to you was largely that most of the energy was being driven by the nuclear genome to synthesize these proteins, and these circles of mitochondrial DNA played a small part. But I've also shown you now that it's not one way, and that bits of mitochondrial DNA are continuously going in to your nuclear genome, even as you sit here today. And sometimes this can be extraordinary. This is just from one cancer that Wei studied, where the mitochondrial genome was smashed into many bits and found scattered throughout the human genome itself. So, back to the opening slide, mitigating mitochondrial mutational meltdown. Can we save the species? Well, hopefully I've convinced you that this is a highly dynamic situation, and the new techniques that we've got are casting more light on increasing complexity. But some of the single-cell approaches are giving us a handle on mechanism, which I believe will lead us to some treatments for people who are affected by these. And that might be relevant not just for the rare inherited diseases, but also more common conditions. Can we save the species? No idea, really. If you invite me back in a few years' time, I might be able to give you a better answer. And I'll stop by thanking the many people that have worked with me, particularly in red, the individuals who've contributed to this work I've described today. Rita, Yele, and Nick Jones, who are strong collaborators, kindly funded in all cases by the Wellcome Trust and other funders, and some of the other collaborators over the years. Thank you for your attention.